Hello.
How's it going?
All right.
I am new.
Okay. Yeah, I see that. So, how are you? Why don't you introduce yourself?
Sure. My name is Paul Mandal. Dual degreed in computer engineering and applied math.
Okay.
Uh, have a master's in cyber, master's in computer science. Most of my research is in AI and machine learning. But right now I'm looking at moving over and doing an MD PhD,
Okay.
and I want to do a lot of I guess a lot of computational biology and neurology research. That's
Okay.
why I'm attending the meeting.
Yeah, that's great. Well, thank you for attending.
Of
And
course.
yeah, I'm not sure who's going to be here today, but usually we have a pretty good set of things that we do here. So yeah, let's see. Usually, yeah, we usually have more people, but like for now, we'll just start. And here we go. There's Lalith. Hello, Lalith. Hi, Lilith. How are you? I don't know if you wanted to give an update or not. Okay, why don't we start? Yeah, hi. There's a tick. Hello.
Good morning.
Morning.
Good. Lalith's here.
Yeah, yeah.
Lalith is with a hard or soft A. Lalith or Lalith?
Okay. Oh, wait, yeah, he's.
Hello, Paul. Where'd you come from?
I'm new. My background's in CS, but I want to do computational biology stuff. So
I didn't have my sound up. Okay, what's your background in?
computer science, primarily AI and ML, but I'm looking at moving over to computational neuroscience. So.
Computational. Okay. Okay. Good. Then we'll have a model for the brain.
So yeah, welcome to the meeting. And today we'll go over a couple things. I'm going to go over some papers that I've been sent, and then an article on OpenWorm, which would be interesting. We talked about it a little bit in the Slack. So this is I wanted to go over this real quick. This is a recent meeting that was held in France at the beginning of June. This is called Shaping Life 3, and it's a very splashy program, and it's basically about developmental biology. So they have a number of different talks here. I mean, you're not going to get much more than that, I don't think, because I don't think it was online or recorded. So, there are keynote lectures, there are other lectures. So the keynote lecture here, the first one was Shaping the Maternal Inheritance by the Cytoskeleton. Then they had this session 1, New Tools and Technologies. They had this invited speaker, Visualizing Morphogenesis Across Modalities. Another talk, Compressive Stress Drives Morphogenetic Apoptosis through Cell Lateral Tension and piezo-1 activity. So, a good amount of things on mechanics of morphogenesis and different ways to deal with that. This other talk, chromatin mechanics at the onset of mouse zygotic genome activation. So, some genomic stuff in there. This one, highly dynamic mechanical transitions and embryonic cell populations during Drosophila gastrulation. So another mechanics paper or another mechanics talk.
Bradley, can you put the URL in the chat for that?
Yeah, I don't know if I have the URL.
Or at least the title of the conference.
Yeah, it was, Shaping Life 3.
Okay.
This was held in France, Cassis,
Yes,
France.
They didn't
Yeah.
solve it in the first two conferences.
Yeah. So then there was Morphogenesis 1, which is the second session. There was neuronal dendritic morphogenesis, mechanosensing in epithelial tissue in vivo, endoplasmic reticulum networks, and retinoic acid signaling to the myocardium drives morphogenesis. So a lot of molecular stuff, mechanics, and other things. Then they had this session 3, Evo, Devo, and Emerging Models 1. This was, let's see, if you're familiar with Evo, Devo, kind of the topics that they like to hit. They had this one talk here, embryonic development of a distributed nervous system in Octopus vulgaris. So that was kind of talking about the nerve net of the octopus. How a single cell shapes a shoot. This is, I believe, is plant morphogenesis. Evolution of temporal patterning of neural stem cells. Again, the sort of differentiation in the brain. Squaring the hexagon cellular and molecular mechanisms, mediating square cell packing in the crustacean.
Oh, that's wild.
Yeah. So yeah, we talked about that last week, about the shape of cells, and actually, we talked about why people use hexagons in modeling, but there is an issue of like why are cells a certain shape as well.
Synod has an illustration, I think from the 1930s, of a cross section of a plant in his book Plant Morphogenesis, which is square. The cells are arranged in a square array.
Okay.
Okay. I'll put the Synod. There.
All right. Oh yeah, Plant Morphogenesis. Yeah.
It's a very good book.
Okay.
And good morning, Hussain.
Hi. Hello.
Hi.
Hi.
Yeah.
Hey. How are you? Nice to meet you.
All right. And then, so of course they have methods interspersed in a lot of these conferences. So this was sponsored by Nikon. This one, intelligent and automated microscopy to extract sample behavior in one click. So always a lot of things on like how to make microscopy better. So then there was Morphogenesis 2, which was the session 4. And again, from embryo shape to the onset of zygotic gene transcription. So you had molecular studies. This one, fluid or elastic. Direct measurements of material properties unveil the early embryonic mechanical response to morphogenetic forces. So the sort of soft materials and material science applications to developmental biology. And of course, you had morphogenesis in Drosophila wing disc, and this one, osmotic control of mammalian embryo size. So again, more things to do with size and shape regulation and development. And then, of course, they had the session Gene Regulation 1, which involved a lot of molecular work, as you might imagine, involving embryonic patterning and epithelial morphogenesis, and doing things like spatial transcriptomics and other types of molecular studies. Then there is physiology and metabolism, which is always a nice divergence from molecular methods. This one had a couple of talks here. One was adaptive morphogenesis of a cellular trap in the developing vertebrate heart. This one was a multicellular actin star network underpins epithelial organization and connectivity. So a lot of things with respect to the sort of, the sort of components of the cell and components of the tissue here. Then there's Evo-Devo and emerging models too. More of this sort of Evo-Devo approach, which is, of course, the evolution of development. So you're talking about, it's not always strictly about evolution. A lot of times it's about diversity and how that arises in evolution. So this one here, divergent evolutionary strategies preempt collision between expanding tissues during flygastrulation. So you have different evolutionary strategies. This is multicellular developmental diversity at the root of animals, and a model for bilateral symmetry, and then exploring the links between tissue morphology and healing, and a spatial model of cell-fate choice. And then, of course, there was a session on stem cell lineage and regeneration. That's sort of regenerative medicine, and a lot of things to do with like cancer research and thinking about how do you model cell fate transitions and using things like organoids and gastroloids now. So this is definitely this one here, invited speaker, why some worms regenerate and others cannot. So if you're familiar with some of the worm models that we have, of course nematodes, and then we have flatworms instead of roundworms. Nematodes are roundworms, and then they're flatworms. And flatworms, in contrast to nematodes, are famous for regeneration. And this is the system that Mike 11 works with, for example. This is the one where the system where you can take a single cell from a flatworm and regenerate an entire flatworm from that single cell. It's amazing. Sort of,
got a question about the nematodes.
Yeah.
I came across a paper recently where somebody cut a developing nematode in half.
Yeah.
And then each half developed mosaically. So it was different. Each half was different. Do you know if one can stick those halves together again?
I don't know about that. Well, I mean, that would be interesting, but I don't know. Yeah. I shouldn't say that all nematodes can't regenerate. It's just like in general, the flatworm is the model system for that. Yeah, I don't know. That's interesting.
Okay.
Yeah, if you could send me that paper, it'd be interesting.
If I come across it again, yeah. Okay. Look for it.
Yeah. So that, and then there's this question, of course, about regeneration. Why do some organisms, why do they have this regenerative capacity and why others don't? And of course, if we could figure out what causes regeneration, we can replicate that, say, in humans or in other organisms to see if we can figure out how to leverage regenerative capacity.
Why are we like nematodes?
Yeah, well, again, we have some regenerative capacity, but not nearly as much as people would like, right? It's yeah, you can regrow your fingernails, but not your spleen or your heart, or you can regenerate some of your heart, but not enough to save you in a lot of cases. Okay.
I'm sorry to interrupt. Did this conference have any published proceedings, or was it just talks?
I think this was just talks, so they don't.
Okay. I was trying to pull up the article, hence why I asked, and I couldn't.
Oh, I put the URL in the chat.
Yeah.
maybe you can check it out, see if they published anything.
Oh yeah, no, I'm looking by the author now, but I tried the title and nothing came up.
Okay. Yeah, they usually don't. Not in the biology conferences. Usually don't publish a proceedings with it as much.
Yeah, try. Yeah, you can try these reference sources to look up.
Yeah, I'm on Google Scholar right now.
So yeah, so then there was gene regulation 2, which of course is more. It's there's an intersection between the gene regulation topics and Evo-Devo, because Evo-Devo is really a lot of it is about how do you how does gene regulation evolve over across phylogeny. So yeah, you'll see a lot of kind of overlap there. This is conserved eye muscle program through development and evolution, for example. And this one, comprehensive analysis of 3' UTR changes as an active process during zebrafish early developments. This is about DNA regulation, and then there are a couple other talks as well. And so that was the last session. There was a final keynote, understanding fruit flies in context, toxins, AI, and behavior. Again, there's this focus on merging biology with artificial intelligence methods, and gaining understanding from that. So it's a very hot area now. Every time you turn
Bradley,
around, yeah,
has anyone done a paper on how old Evo-Devo is now?
Well, yeah, I don't know. Seems like an interesting topic. I guess it kind of came together in I don't know, the 80s, maybe.
That
90s,
late?
maybe the
I thought it was
90s.
earlier.
Well, there were studies that were published, and you had some landmark studies. I don't know where you demarcate the beginning of a field, but the first mention of it maybe would be the place to go. But I don't know, maybe the first mention of it might be as late as the 90s. I'm not really sure, but it might be an interesting thing. Yeah.
Yeah, it seems to me that its promise has not been realized.
Yeah. Yeah. Some people argue that it's not a theory, that it doesn't really function like a traditional theory. And you think about a theory, it's supposed to, give some new set of explanations, and it's kind of oh yeah, development happens in evolution too. So, but in any case, yeah, I mean, it'd be interesting to see, and that's something that I know that is pretty
Okay, Grok
special.
claims it started in the 1970s.
Okay, yeah.
I'm sure those questions can be evaluated in terms of the power of the theory, looking at potentially explanatory, predictive, and other forms of theoretical advantage that it might give. One common bottleneck for many potential scientific theories is they may be descriptive, but they're not so predictive.
Yeah.
But it's interesting. It's interesting. Yeah. The predictive power of those theories would give it potential to argue that it is a theory. It's not just some random explanation that we don't need. So
yeah. Okay. Yeah. So thank you. So before we go on, I don't know if Hussain wanted to give an update on his work. I mean, I know you gave one last week, but.
Oh, sure. So once again, we're working on plenty of stuff. Evo's Evo. Cool.
Yeah, I put the Grok's view on it in the chat.
Don't trust Grok. Don't trust Grok.
Oh, I know that.
It's far less reliable than ChatGPT and the others.
Oh, really?
In my opinion,
even the better models of ChatGPT, like the ones that use RAG, they still hallucinate references. It's horrible.
Oh, Grok blatantly blatantly biased. Grok just has its own problems with that. Anyways, okay, excuse me. Yeah, so working on a couple different projects alongside Dick right now. Mostly the CT project, the ray-by-ray computed tomography project, but also the peptide project on the origin of peptides through simulation, how they've been formed over whether or not we can describe the statistical mechanical model for the lipids first hypothesis and simulate the early peptide formation and division using these 2D hexagonal grids, which we had talked about earlier. Okay?
Yeah.
So I'm still working on that, getting the figures and the images together and consolidating a lot of the writing that Dick had mentioned earlier. One important point that Dick had pointed out was that I think that the excuse me, it seemed like Dick had raised this argument that the what was it, that the alpha helices, the one particular hypothesis you mentioned doesn't hold so much water. Is that correct? Was it about the alpha helices? Alpha helices, that one?
No, it was the problem is with the RNAs.
Oh right, it was the RNA.
RNA precursors have been detected in meteorites, but nobody's figured out whether they have chirality.
Okay, yeah, that was the, and we had proposed looking at the racemic.
Yeah, it looks like the empirical basis for going after them may not be there.
Okay. Well, that's important to point out, and I appreciate you mentioning that. Yeah. So just to back up for a bit. Once again, this is the project that Dick and I are working on with the colleagues over in Italy, with Savino and his colleagues Gaia and Grazia.
Yeah.
And
Well, the basic idea is that the alpha helix is hydrophobic, and it can be fairly short, so it can anchor a peptide in the membrane, which is hydrophobic.
yeah. Okay.
And then there's a positive feedback which we think should occur. We have to simulate it carefully, and that is hydrophobic mismatch, where the length of the alpha helix is not the same as the length of the amphiphils that make up the meteorite vesicles. The meteorite vesicles have been demonstrated, but nobody's actually analyzed them, as far as I know. However, the components of them are fairly well known. And I just came across something interesting. They seem to be dicarboxylic acids, which means that both ends are charged for the molecules in the membranes. And that's the same structure as you find in archaea. Okay, both ends charged, and you don't get a bilayer, you get a monolayer, but with both ends charged of the same thickness.
Okay. Yeah, so that's okay, that's interesting. And okay. And that's how they have the de novo fatty acid synthesis, right?
Yeah. Deletion
of the N-oil carboxylic acid reductase gene. Okay.
Well, we're assuming there are no enzymes around or genes or anything like that. So the process we're trying to simulate is strictly physical chemical.
Okay.
And the energy source is one that's generally ignored by people who are worried about energy, and that is just the rotation of the Earth.
Okay.
Which presumably can drive temperature or wet-dry cycles, and therefore lead to polymerization of peptides.
Okay, yeah, I'm glad you're being thorough with which hypotheses have been explored and which ones haven't. So, yeah, exciting stuff.
Yeah, I mean, some of these are experimentally approachable, but nobody's not.
Cool stuff.
So if you want to trade in your computer for a test tube, go ahead.
Yeah, speaking of which, did you guys appreciate that article I'd sent about the worm in Wired?
Oh, the one in the Slack that you
Yeah,
posted? Yeah,
Somebody
we'll
in one of
talk
the
about that in a little
crashed
bit. Yeah.
the Open Worm project, saying that the Open Worm project, despite being a decade plus, has been an absolute failure. And that's
Oh,
wow.
really?
Okay.
Failure at what?
Yeah. Well, I mean, I read it a little bit differently, but I mean, I know that like they.
Yeah, I don't think the article was quite that vindictive. I do remember when the open worm project came out, though. It's like, oh, this is a small worm. We can gain a full understanding over the next 5 to 10 years, and you know things went very differently.
Yeah.
They literally start off by saying the project for 13 years a project has tried and utterly failed to simulate it. You can say they're trying to present the challenges about it, but it sounds like they're just like bashing on it, you know.
Didn't Tom Porteous make a robot that acted like a worm?
Yeah. Yeah. So I mean, yeah, a couple words about that. Well,
Where's
maybe
the
we'll
other
get into
fellow?
the article now, but yeah, a couple words on that. The first one is that we talked about an article several months ago, maybe, where they had kind of the same claim that like any kind of simulation is of and then they talked about open worm in there that any kind of simulation has failed, and then like kind of talking about how to how they could do it better, and how it's kind of this is a way to do it, but I don't think that their way would
There have been
necessarily
tons
work
of
any better.
roadblocks, tons of bottlenecks, tons of
Right.
challenges. But I'd say it's very, very far from a failure. You want a failure, you look at like
Right.
the Human Brain Project or something like that. Like
Yeah.
that's been a train wreck for years.
Well,
Even
we
Yeah,
we talked
I've
about that.
been trying to get the number of different kinds of cells in brains, and there's almost no literature on it.
Yeah, it's yeah, it's a thing about like a lot of these projects, and we talked about the Human Brain Project as being like if you're aiming for like simulating or emulating a biological system, I mean there are a lot of ways you can fail at that goal, but you produce a lot of things underneath it. Even if it's simulation, you produce a lot of potentially useful things. And like at the Human Brain Project, they produced a lot of things that help people, doing brain science or simulation or whatever. And so it's not in that sense, it's not a failure. And of course, you can do these simulations and you still learn things. You still build sort of these you test to see if models work to simulate some aspect of the process. So like yeah, Tom Porteous has been working on like building C. Elegans inspired robots. He's been working on other things. He's kind of working on this old school artificial intelligence where he that was I think what he did his PhD dissertation on, and it's just like this kind of cottage industry of his where he's simulating different types of intelligence and things like that. So you're learning things about how to do simulations. You're learning things about the phenomena itself. So it's
Yeah.
interesting in that way. I
Yeah.
mean, yeah,
It
go
also
ahead.
needs some perspective. Those of us who were born before the year 2000 are quite aware that for decades, artificial intelligence was regarded as a total failure.
Right.
yeah, it's honestly up until ChatGPT, people have said it's just an absolute failure. You know.
Yeah.
Nobody cared about AI making pictures of people if they look like trash up until this point. So
Yeah. Well, I mean, yeah. So why don't we go to this article?
I appreciate that but once again, changing the tone here, rather than some like straight up cynical doomerism that it's all a fatalistic project that hasn't worked out. It's more so like a holy grail type thing. Yeah.
Yeah. I think.
Perfect.
I don't think a lot of outsiders who don't do research understand that science is incremental, and sometimes you're brutally fighting to just gain an inch in your research area, right? And it's clear that the author doesn't understand that because they even say at the very end, that like
Well,
oh they're
the author, she's a tech writer. She's written about
Yeah.
tech and things. So she knows about computers. She knows how computers work. She's not just saying like how come we can't just tell the computer like, hey, program how a worm wiggles, and then it's just
right. But the question is, are they a researcher? Like keep in mind
I mean, she's
how different.
regardless. I think the you know, and it's talking about who wrote it specifically. I think that the you know excuse me like it's a great article in presenting the challenges. It's a great article in presenting the challenges of the Open Worm project for the past the past decade or so, as they mentioned. But it's like how incremental everything has to be for years and years and over a decade and stuff. Like that's what they're trying to get at, right? So that's why I say it's more like a holy grail analogy rather than just like an absolute like dumpster fire of a project. That's what I mean.
Yeah. Oh yeah. So yeah, let's go to the article here. See if I can pull it up here. All right, yeah, this is the worm that no computer scientist can crack, and you know, it's there's this history actually of different C. elegans based projects, and of course we've talked before about why C. elegans is this thing that people want to simulate. You know, it has a fixed number of cells in the adult. We know what the sort of the lineage, we know the full lineage tree, we know what every cell will become. So it's both deterministic in terms of cell differentiation, and it's what we call eutelic, or it has the same number of cells in the adult. Like, you know, every hermaphrodite has the same number of cells, every male has the same number of cells. And we know what developmental cells will become what adult cells, so we can simulate it cell by cell. And so that, and then, you know, part of the problem here is that that's just the cells. And then you have to simulate the connections in the connectome. You have to simulate the molecular milieu inside the cell. And we know we have pretty good genetics of C. elegans. We know how, you know, we have the full genome sequence. We have a lot of defined mutants, so we know what genes actually do in a lot of cases. But still, there's a lot of biology there, you know? And so it's like you can simulate it at different levels of understanding. And, you know, you can produce like an output, which is like a worm moving around in a simulation that looks a lot like a worm that is in real life. But then you still have this problem of like, is it producing it through exactly the same means? Or is it just kind of a similar looking process, but it's producing it differently because you're not modeling everything that goes into a worm movement? You know, so there are questions like that. And you know, it's fine, but there have been, because of that sort of simplicity, there have been a lot of projects that have come and gone, going back to the 1990s. And so there's this history here of like, you know, people kind of taking that bite and then, you know, kind of not producing anything. I mean, maybe they produce a paper or something. So in that sense, OpenWorm hasn't failed at all, because OpenWorm has kind of started off, and you know, there have been a number of different software projects, open source software projects, that have kind of resulted. And then what they refer to in the article about running the worm, it's basically a program that runs in a virtual machine, that you can run all the programs at once and get an output. So you can run, you know, cybernetic, which is the biophysics, the neuron simulations, and other things, and then run those and get an output. So it's interesting that, you know, and then that's kind of where we've kind of been for a while. You know, the goal, of course, would be to merge all that together, into a unified simulation that works. So that's kind of the issue here. I mean, you know, I guess it's not necessarily an issue. It's just that, like, if you're looking for an easy to run simulation of the worm, you can see that, like, that may or may not be like a viable goal at this point or maybe any other point. We have to kind of take steps to get there. We can't just do that in like 10 years to get to the full sort of an emulation of a worm. And if you wanted to do an emulation of an organism, I mean, you could do single cell simulations or single cell organisms. So we talked about in this group about some of the initiatives people have done with bacteria, like modeling, like maybe like a bacterial cell with a minimal, very small genome, and then how much that takes. So like there are a number of groups modeling whole cells, and and these are like bacterial cells that where one cell is the entire organism, and and to do that they do a lot of modeling of the genome, modeling of the proteome, modeling of the cell morphology, and just with that one cell, it takes massive amounts of computational power to generate like molecular dynamic simulations, maybe at the scale of a couple of seconds. So there's a
Yep.
lot of, if you're
Yeah, like
dealing,
they mentioned they repeat that one quite a bit. 10 hours of compute time
yeah,
for 5 seconds of an animation.
yeah. I should,
I mean, if you ever use Blender, that's how long it can take at times too. I think I made like a 20-second animation of a donut spinning, and I think it took my laptop like 2 hours to render.
yeah. Yeah, it does take a long, especially when you get into these.
The donut tutorial from Blender.
Yeah.
Yeah, the progress on computers is incredible. When I first dealt with image processing, the only digital images I could find were pictures of clouds taken by Noah, which were very hard to work with. No sharp edges, nothing. Okay. And then somebody came out with a movie of showing a face citing the poem "How Now, Brown Cow."
Yeah, yeah,
And they pointed out it took them 45 minutes per frame to generate the movie.
yeah.
Okay.
I remember that one. There's the Ghibli scene. This one Studio Ghibli movie that is like 3 seconds of a crowded like shop mall type place. So a lot of people walking in many different directions in an outdoor like market type shop.
Yeah.
I think that entire 3 seconds of animation took multiple weeks for their studio to make. Yeah.
Yeah, I mean, computers are so much faster now.
That was all hand drawn. That's the one I was thinking of. But
yeah.
I think the article does do a lot of good merits for challenges and struggles for a very long time. Okay, I really appreciate that. I think it's really solid with pointing out, from a more objective manner, the good sides, the bad sides of these things, and really putting into perspective how difficult it is when you say I'm simulating a worm on my computer, and how difficult that really is from biologically and scientifically realistic perspective. So,
Yeah. I should prepare. Oh, go ahead.
Naive question about worms. Do the nerve cells generate spike trains?
Not in the way that human neurons do or mammalian neurons. So they generate like a sort of a semi-action potential. I guess they generate something like spike trains, but yeah, it's a little bit different than what we know in.
Okay, so
yeah.
even that has to be modeled. You can't just
Oh,
assume.
well, the OpenWorm has actually made some pretty good progress on doing stuff like that. Yeah, I mean, like,
Oh, okay.
yeah, because
Okay.
it's really a large component of it is the neuroinformatics aspect, because we have this connection with INCF. So a lot of the neuronal stuff that's like a core kind of thing that is of interest. So like one of the reasons why DevoWorm started was because they didn't really have any good account of development. They had a lot of stuff on connectomics and the nervous system, but not necessarily on development. So
Okay, so it's really been 13 years since we started this.
about 10 and a half. Oh, OpenWorm is been about 13 years. Yeah.
Yeah. Okay.
Yeah. Okay. So yeah, well, like I said, though, I should prepare like a little chart to show like the molecular dynamic simulations and their computational power requirements and their time simulated, and then compare that with like some of the OpenWorm stuff that you can produce in a virtual machine on your laptop, which is interesting because, I don't know if there are a lot of containerized molecular dynamic simulations. I imagine there probably are, but like just showing that you need to have, and
Yeah,
especially
there are.
depending on
Like
what you're
there's yeah,
trying to
because
simulate, a lot of power for that.
I'd like to see you make the jump into virtual research as well too. So, these articles in the science communication pop sci community, they're nice. They're nice like reminders of what we're doing, like to communicate those things to the general audience so they can understand this stuff too. So, yeah, it is. It's it is what it is. Cool stuff. So, yeah.
Yeah.
Like I think the biggest remember this is all completely live, like open source, right? And
Yeah.
I feel like there might
Yeah.
have just been this idea that if we just collectively gather the power of anybody on the planet who wants to work on this can do it, right? We should be able to get it done as quickly as possible. And then vastly underestimating how difficult that is too in many ways. Conducting open source can be pretty confusing at times. So like an open source research project or just like a simulation type thing, it takes a lot of coordinated effort.
Yeah.
So, it is cool though. It is cool though, and I appreciate that. Like, at least compared to many other open source efforts in science and research, like from my background in bioinformatics and computational biology, a lot of the open source projects they would make and do in there still felt pretty like closed off and guarded by the academic community, and I felt like they were never nearly as responsive to new data, new results, and new power as quickly as they could be, because people just gate kept the hell out of their. And it was just like pretty simple like gene splicing and annotation software
Right.
that would never update itself as quickly as it could with computational power as a whole for technology and software. So yeah.
Yeah.
Anyways, this is I do overall appreciate the article. I do. So yeah.
All right. Yeah. So this was published in Wired magazine. This was, I don't know. I mean, it's online, so it's the worm that no computer scientist can crack. Yeah, it had a lot. It was a lot of interesting things here. And they talk about Stephen Larson and Jorge Gleason, and these are two of the people from the project, two of the senior contributors. They also talk about Netta Cohen, who's on the scientific advisory board, and she does a lot of interesting work in like doing C. elegans wet lab work, but also computational work. It's interesting. A lot of the things here. Um, yeah, so it has a lot of the points here that we talked about. Someone said here, who's Haspel? Gal Haspel, a computational neuroscientist at the New Jersey Institute of Technology, and the lead author on the reverse engineering paper. This is that paper that was published recently where they proposed taking this kind of new approach to C. elegans, where you go back and you do experiments and you bootstrap some sort of total simulation from that. So another attempt at this sort of thing. Um, and he says estimates that pulling it off, it being this simulation of total simulation or emulation of C. elegans, may take up to 10 years, cost tens of millions of dollars, and require something in the neighborhood of 100,000 to 200,000 real life worms. So this is like where to do simulations, you need to do wet lab biology. And C. elegans or in OpenWorm has relied upon secondary data sets, different C. elegans labs. And C. elegans is nice because you have a lot of labs that are devoted to doing things in C. elegans, and there's this nice community where they share their data, and they share their reagents, and they share their defined mutants. So you have access to a lot of these things across labs. But what they're proposing in this reverse engineering paper is to sort of build the infrastructure up kind of from scratch, and then do these mass experiments where you're collecting lots and lots of data and then training the model. And you can actually do very controlled experiments relevant to the simulation. So it's very much connected to that, instead of just kind of, getting as much data about the worm and then kind of running a general simulation. But of course, that also means that costs a lot of money and takes a lot of time. So in the process of this, it would generate more data about C. elegans than has been collected in all of science to date. And what in the end will the reverse engineers have to show for it? All these people and all these computers, Haspel said, and will end up doing what one little animal can do right now. He compared that project to a NASA moonshot. It's the kind of undertaking that drives technology forward, pushing engineers to build better tools and scientists to work together. And so this is where, if you combine enough automation, big data, and machine learning, you get this simulation or emulation of an organism. So this is kind of going through a lot of this sort of this whole area of how do you simulate an organism. And again, C. elegans, the reason people are interested in that, and again, we've done like these single cell simulations that are very detailed at the molecular scale and are single cell organisms, so they're not particularly interesting in terms of behavior or even physiology, a lot of it. And that's why we want to do C. elegans, because C. elegans, aside from being simple, which it's not, but it's simple enough to get a handle on, you still get these interesting behaviors and these interesting sort of things going on in development and in in the life history of the thing, physiology. Yeah. So that's I think we'll move on from that paper now. I think that was a good kind of discussion. Thank you to everyone who had some thoughts on that. We'll continue to discuss it in the Slack. It's in the DevoWorm channel of the OpenWorm Slack. So if you want to have more conversation on that, that's the place to go. I did want to kind of go over, before we go, a couple of papers that I've kind of run across that were sent to me, etc. This one was we talked, well, I think last week and the week before, about mechanical aspects of development. This is a nice paper on control of epiblast sulfate by mechanical cues. So this might be of interest to people like Susan. And this is where they're looking at embryonic tissues originating from multipotent epiblast cells arranged in a mosaic of presumptive territory. So they're looking at like how tissues differentiate, but they differentiate spatially into different locations in the emerging organism. This is a mammalian. This is an amniotes. So this is not in C. elegans or in Drosophila, but this is in amniotes. So that's a portion of the vertebrate phylogeny. How these domains faded to specific lineages, become segregated during body formation, remains poorly understood. Using single-cell RNA sequencing analysis and lineage tracing in the chicken embryo, so they use chicken as their model organism, we identify epiblast cells contributing descendants to the neural tube, somites, and lateral plate after completion of gastrulation. We show that intercalation after cell division generates important movements of epiblast cells, which lead to their relocation to different presumptive territories, explaining this broad spectrum of fates. So this is combining sort of cell differentiation and migration, and then the mechanics of these cell populations, the sheets moving around, folding, doing things like that. This tissue remodeling phase is transient, being soon followed by the establishment of boundaries restricting cell movements, therefore defining the presumptive territories of the epiblast. Finally, we find that epiblast faces distinct mechanical constraints along the anterior-posterior axis, or the AP axis, leading to cell fate alterations when challenged. Together, we demonstrate the critical role of mechanical cues in epiblast fate determination. So this is, this is again is in amniotes. So development is a bit different than how it proceeds in C. elegans or even in Drosophila. But the take-home message of this paper is that you have this sort of spatial aspect to development, you have this mechanical aspect to development, and you have this aspect of organizing cells in sheets and in sort of these the shape or these geometric structures that then determine a lot of what happens. And so, we see these things in development, but we can manipulate them as well and get interesting outcomes. So that's, if we're interested in cell mechanics and in the organization of cells into these larger structures, that's an interesting paper. This is a paper on active nematics. We talked about nematics back, I think maybe 2 meetings ago. This has to do with how cells are oriented in development. So are they oriented all in different directions in terms of the sort of the way in which they move, the way in which maybe their oblong shape is oriented to one another. And so if it's smectic versus nematic, and so you have these different, in nematic tissues, everything is oriented in the same direction, and that has consequences for how development proceeds. And you can think about cells is engaging in this collective behavior where maybe they all move in the same direction during migration, or they're all oriented in the same direction so that they go on to form things like muscle fibers, or things related to anatomical orientation. So this is kind of thinking about this maybe larger scale physics than single cell physics, and how that plays a role in development and shaping phenotype. And so this has this author here, Matthias Serra. This person, I think, has done a lot of stuff on Lagrangian coherent structures, which is an interesting approach to physics where you look at flows and you try to predict these meta structures that exist in flows. And you can find Lagrangian coherent structures in flows where they form these ridges, and you can calculate from certain origin point to the edges of these ridges, and you can say things about maybe the collective migration of cells, or in a lot of physics applications you can say things about the flow itself. What is the flow? The flow, it has maybe a certain degree of turbulence or laminarity by the way in which the things that are moving in that flow and where they end up. So if you have this a ridge with a very complex shape, you can say that that flow is turbulent, and there are a lot of things you can do with that tool. In this case, they're looking at material transport and fluid flows, and they're looking at these objective Eulerian coherent structures. And so, they were talking about this in terms of active materials. So it's not necessarily related directly to development, but they say that there's an application domain in terms of synthetic active matter and morphogenesis. So if you're doing things like modeling, cellular structures, I know we've talked a lot about organoids. There may be applications to organoids or to morphogenesis in actual organisms. And so they talk about how objective Eulerian coherent structures and instantaneous Lyapunov exponents govern short-term material transport and fluid flows. And then they talk about these Lagrangian coherent structures. And then the way to measure that is through this finite time Lyapunov exponent. And so, you use that exponent to describe these coherent structures, and that describes something about the flow that governs, say, like migration or movement of things over time. So, and then of course you have this Eulerian rate of strain tensor, which is something we can also use to understand these flows. So here we devised for the first time an optimal control strategy to create short time attractors and compressible viscosity dominated active pneumatic flows. So this is again having to do with treating development and cell migration and things like that as these compressive viscosity dominated flows. And so you can actually do this to model how things are going to behave in development or in some sort of cellular system. By modulating the active stress intensity, our framework achieves a target profile of the minimum eigenvalue of the rate of strain tensor, controlling the location and shape of these short time attractors. So they're using dynamical systems theory to predict the location and shape of these sources of maybe like these ridges that I discussed. Those are these short time attractors. So the ridges kind of move over time. You're measuring from an origin point to the edge of a ridge at some point in time, and that ridge changes shape and moves away from the origin point, or maybe even towards the origin point over time. And you're looking at the dynamics of that system. So you want to have some account of order in the things that are migrating, the things that are moving around. And so that's how you can describe them through these ridges or these short time attractors. We show that our optimal control strategy effectively achieves desired short time attractors while rejecting disturbances, meaning that you're getting the actual signal instead of artifacts. Combining optimal control and coherent structures, our work offers a new perspective to steer material transport in compressible active pneumatics with applications to morphogenesis in synthetic active matter. So this is a pretty hardcore physics paper, but it does have some applications to biology and development. So they're interested in these large-scale coherent dynamics where global collective behaviors arise from local interactions, as well as describing individual anisotropies and different types of ubiquitous activity in such systems. So what kinds of systems are we thinking of? Well, like I said, we have developmental systems, but we can also use this type of methodology to describe bird flocks, so collective behavior of agents, bird flocks or cells, in these kind of computational models. Sometimes we can describe the movement of bird flocks or the movement of cells, and we don't have to distinguish between the things that drive them, or like the brains in birds and the sort of the uh, biochemical or kinetic pathways in cells, because they produce very similar behaviors, and we just simulate the things that lead to those behaviors or analyze the things that the outcomes of those behaviors. We can also model bacterial swarms or ensembles of cells that exhibit macroscopic patterns whose length scale is orders of magnitude larger than the individual size. So, this kind of goes into some of the biological domains that we can use. We can use this method with, and the paper gets into some of the theoretical methods that exist now and how this improves upon it. Okay, this last paper here I wanted to highlight is this PNAS paper, and this is dynamic forces drive cell and organ morphology changes during embryonic development. And again, this is more about mechanics and about how we can, develop models to understand these mechanical forces and some of these other aspects of cell migration. So the abstract here reads, cells, tissues, and organs must change shape in precise ways during embryonic development to execute their functions. So we're dealing with shape and shape changes both in cells and then in tissues, which are collections of cells, and then organs, which are collections of tissues. So we're dealing with these three different scales. And so we need to understand not only that those different scales, but then the changes in shape and how to analyze that and understand. And of course, those shape changes involve multiple mechanisms. So biochemical signaling pathways and biophysical forces play a large role in that. So if we model those things, we can understand, we can have a working model of how those shape changes work and how to predict them in different systems. It's been difficult, however, to tease apart the contributions of each, especially from tissue scale dynamic forces that are typically ignored. So these are, this is where you have things happening at multiple scales. You have dynamic forces at the tissue scale that affect the cellular scale. But how that works is hard to understand because tissue scale dynamic forces are collective phenomena, depends on the cells contributing to the tissue. So it's hard to get to tease all that out and build an effective predictive model. We use a combination of mathematical models and in vivo experiments to study a simple organ in the zebrafish embryo called Kupfer's vesicle, or KV. Modeling indicates that dynamic forces generated by tissue movements in the embryo produce shape changes in KV that are observed during development. Laser ablations in the zebrafish embryo that alter these forces result in altered organ shapes matching model predictions. These results demonstrate that dynamic forces sculpt cell and organ shape during embryo development. So this is, again, a very key question in developmental biology. How do organisms robustly control the morphology of cells, tissues, and organs, given that information for such structures is ultimately encoded at the scale of molecules? So you have these things happening at the molecular level are being expressed in cells, then that becomes part of the collective behavior of cells, becomes part of the tissue, and then those tissues interact to form organs. So if we go to the significance, they talk about how we aim to understand the mechanisms that control precise cell and tissue shape changes required for organ function. So it's not enough just to predict shape changes generically. You need to have this sort of model of precision where this change in shape results in this change in function, and to be able to predict different changes in shape and different changes in function, of which ones are the most predictive. So that's a challenge. You can predict, a lot of times you can predict shape changes or what leads to shape changes, but then you have this additional aspect of function. And so that's the thing that they're looking for. Many studies focus on cell shapes, have ignored the role of dynamic forces self-generated by slow tissue flows. So again, we go back to the flows of tissues, flows that we can model using techniques from physics that we often use to model soft active matter and other types of physical phenomena. But recent work showing that tissues are near a jamming transition with diverging relaxation timescales suggests that slow motion could give rise to large forces. So this jamming transition, we know it happens in development, happens in soft active materials, and that is where if you get enough cells or enough particles in a volume of liquid, it can induce this jamming transition, which is a phase transition where that population of cells or particles can go from moving sort of in a laminar flow to jamming up and stopping, and then nothing moves. So you could have a laminar flow where you have a certain density of cells or particles, and then as that density increases, say through cell division, you can end up with this jamming transition where everything freezes up and nothing migrates. And you see this in development. We've talked about a number of papers where you see this jamming transition. And you can have unjamming transitions as well, where things open back up. And so one of the aspects of development, or one of the open problems, is how do these jamming transitions play a role in cell migration, and how does it govern development? So they do this mathematical modeling, imaging, and use these mechanical perturbations to look at the tissue scale dynamic forces sculpting the shape of an epithelial organ in the zebrafish. Because many processes during development occur at similarly slow rates, this suggests that self-generated dynamic forces should be investigated more broadly. And so this is an important point here, is that in development, we have events, sometimes in the same type of system that we're working in, that happen at many different timescales. So we just talked about how, it takes a lot of computational power to model a certain timescale. So if you have more details in your model, the shorter timescale you can really afford to have to model something precisely. So if we have these slow timescales, it's going to be hard to incorporate everything from the molecular scale up to the organ scale, and get an effective or even any sort of coherent simulation out of it. So I thought that was another interesting point that they made. So I wanted to go through those 3 papers. I think those maybe kind of highlight some of the things we've been talking about in recent times. So we have some things in the chat. Let me go over those. Let's see. Let's go up to the top here. So yeah, we talked about synapse plant morphogenesis. We have a new paper on shape. This is Dick's paper. Is there a relationship between embryonic electric currents and differentiation waves? That's in biosystems. And then Paul said, I think some of the top pulls from this paper. This is
That was in relation to the worm regeneration stuff.
Okay, yeah.
The author who gave the talk was the senior author on that paper.
Oh, good. Yeah, great. Thank you. This is evolutionary dynamics of whole body regeneration across planarian flatworms. Yeah. Oh, that's great. Well, thank you for that. Yeah.
I'm useful sometimes.
Well, good. Thank you. Yeah, Dick had this question here. When did Devo start? And that's something that maybe is interesting from a bibliometric standpoint, and might revisit that. Then Hussain said everyone's just so obsessed with numbers. And then
you were talking about like all the scales, like, oh, this takes 10 hours to do something, it takes a worm to do 5 seconds. It's like, cool, congratulations. What does that tell you?
Yeah, definitely. Yeah, this is also from Hussain. I have the option of taking CSE 6742.
Oh yeah, a few courses in my master's program that I can now take electives for in the next coming semesters, the next year or so. And I think one of them even used PyNetLogo at one point to NetLogo. Remember we used that for the GSOC project.
Oh yeah.
NetLogo or PyNetLogo. But our master's program capstone at the end involves like a practicum, which is basically more or less like a self-run student project that we get a full project course grade and stuff and things for. So
yeah. So this is what program are you in, by the way?
OMSA, OMSA, the Online Masters of Science in Analytics through Georgia Tech, Georgia Institute of Technology.
Okay. Yeah, that's great. Well, thank you.
Yeah. I had a class this past semester. It was really annoying. I had to take 4 proctored exams in this past summer. It's just annoying. It's just annoying. I feel like they all know that the intro class is just hurdles you have to jump through. So
yeah, after reading about the experience with graduate algorithms, I was like, screw this, I'll do University of Texas.
Yeah, I mean there are different programs like Jesse did UCSD, right? For his master's in data science. Okay. Michigan's really good too. UCSD, Michigan, Texas. Maybe Purdue has one. I'm not so sure. Probably Purdue probably has one too. So Georgia Tech's OMSA is really great because it's completely online, and you just have to take about 10 courses at your own pace, and that's basic BS. So, yeah.
All right. Well, I think that's all for today. Thank you for attending, and see you next week. Thank you.
Bye.